First, I want to thank Bob and the McGovern and MIT for hosting this nice symposium, and also thank Ying for the nice introduction. Indeed, I'm the newest member of the McGovern Institute. As Ying just said, I just went back to China about 2 months ago, September, and it's a great honor to share with you my postdoctoral work and a ice tip of what I'm going to do for my new lab. So the title of my talk today is Visualizing AMPA Receptor Synaptic Plasticity in Vivo. So to this audience, we will agree that the human brain is the most complex organ in the universe. The human brain estimate consists of over 100 billion neurons. The neurons connect with each other and so that they can communicate with each other. To this audience, I just briefly introduce that the neurons communicate with each other through a specialized area called synapses. So this figure illustrates how the neurons communicate with each other. Basically, the signal comes from the presynaptic side, which are activated presynaptic neurons, which will trigger the release of the neurotransmitters. The neurotransmitters will transduce across the synaptic cleft and then bind to the postsynaptic receptors. Then the binding of the postsynaptic receptors will trigger a series of signaling cascade. In such a way, the signal is transduced from the presynaptic neuron to the postsynaptic neuron. So for the synapses, actually, the formation and elimination of the synapses and the efficacy of the existing synapses are a very dynamic process and can be modified by human experience. So this dynamic process is called synaptic plasticity. And synaptic plasticity plays a very important role for higher brain functions, such as learning and memory. So in terms of synaptic plasticity, it's best studied in the using electrophysiology method and in the especially hippocampal acute brain slices. So as shown here, here is the recording electrode, and on the either side is a stimulation electrode. So if you record the baseline, you'll see the baseline is relatively stable. But if you pair the recording with a high frequency stimulation, what you can see is you'll detect a dramatic increase in terms of EPSC amplitude, indicating there is an increase in the synaptic strength. While on the other hand, if you pair the recording with a low frequency stimulation, what happens is you'll detect a decrease in the EPSC amplitude, or in other words, you'll detect a weakening of the synaptic connection. As you can see from both those diagrams, this phenomenon is called LTP, or long-term potentiation, and this phenomenon is called LTD, long-term depression. So as from the diagram, you can see another characteristic of LTP and LTD is that they are relatively long-lasting. In the acute brain slices studies can be as stable as half an hour to an hour. So AMPA receptors is the major glutamate receptors located on the postsynaptic side. So AMPA receptors mediate the majority of the fast excitatory synaptic transmission. So here I'm showing you the structure of the AMPA receptors, which are basically tetraheteromeric structure with 3 to 4 transmembrane proteins. And based on their structure at the segment tail, it can be categorized into 2 categories. One is called long tail, the other one is called short tail. As shown here, the segment tail is really important for AMPA receptor trafficking and function, since it has a lot of protein phosphorylation side, protein binding side, and also some interaction with some binding partners. So the trafficking of AMPA receptors into and out of synapses is a very dynamic process. It plays an important role for synaptic plasticity and learning and memory. So here it further categorized the importance of AMPA receptors in mediating synaptic plasticity to be a specific LTP and LTD. So as shown, multiple evidence have shown that the insertion or the incorporation of AMPA receptors from perisynaptic side to the synaptic side can mediate LTP, or long-term potentiation, while the removal of AMPA receptors from the synaptic side out away from the synaptic side can mediate LTD. So it's not surprising that the disruption of AMPA receptors will lead to a lot of human diseases. Indeed, there are genetic links have been found that the disruption of AMPA receptors or the dysregulation of the AMPA receptor function leads to a lot of neurological or neuropsychiatric diseases. For instance, intellectual disability, autism disorder, schizophrenia, Alzheimer's disease, and dementia. So again, due to the importance of AMPA receptors play in synaptic plasticity and overall brain function, so it's a very hot field of the study for the scientific research. So one of the way to study AMPA receptors is through imaging. So here I'm showing you a dissociated neuronal culture. Those are the hippocampus neurons. Here the cell body sending out beautiful neuronal processes. And the magenta shows the cell field to outline the neuronal processes, while those green puncta are the staining of the AMPA receptors, which the puncta pattern is a characteristic of the AMPA receptor allocation. As Jun just briefly introduced, another trick we can use to study AMPA receptors actually is the super ecliptic fluorine. As Jun just mentioned for his talk, that one nice thing about the fluorine tag is that when it's trafficking in the vesicle facing the acidic pH, the fluorescence is quenched. Once the tag is delivered to the membrane surface facing the neutral pH outside the membrane, you'll detect a dramatic increase in terms of fluorescence intensity. So in such a way, using this tag, we were able to basically follow the dynamic or the trafficking of the membrane population of those AMPA receptors. Indeed, previous work from Rick's lab have been shown or have been using this tag to study AMPA receptor trafficking or membrane incision in the dissociated neuronal culture. As we can see here, here's the cell body of a hippocampal culture. What we do is we basically photo bleach and eliminate all the fluorescence on the surface of the on the membrane surface from the AMPA receptors. Then we use turf microscope to observe the insertion of AMPA receptors from the inside of the cell. What you can see here is, take a time series, you can see those bright puncta structure showing up. Basically, those are showing a group of AMPA receptors inserted into the membrane surface. Using this method, we were able to study AMPA receptor trafficking and the dynamics, especially for the membrane population in the dissociated neuronal culture. So over the years, a couple labs in the world, including our lab from the Peking lab, have been identified a AMPA receptor complex that are playing an important role in AMPA receptor trafficking and regulation and synaptic plasticity. Here I'm showing you a very oversimplified version of the AMPA receptor complex. So basically, those are the protein-protein binding partners and kinases that either modify or regulate AMPA receptor trafficking. So as I said, the research over the past decade have dramatically advanced our understanding of AMPA receptor trafficking and regulation, especially at the molecular level. However, a major missing link in the field is we are still unclear about how AMPA receptor trafficking and regulation are related to animal behavior, or how does AMPA receptor traffic and behave in vivo. So when I joined Rick's lab way back some years ago, we decided to address this question using two-photon in vivo imaging. So here, this slide shows you the basic strategy for how we are going to image AMPA receptors in vivo. So first, we are going to deliver the CEP-tagged AMPA receptors into live animals using a method called in utero electroporation, which I'll explain a little bit more about this technique next slide. And then the second thing we need to do is to do a craniotomy, basically as to either implant a window or thin the skull to, so that the microscope can image through to observe AMPA receptor signals. Of course, the most important step is to really image AMPA receptor dynamics in the live animals under certain synaptic plasticity conditions. So this slide shows how in utero electroporation works. So in utero electroporation was pioneered by a Japanese group about 15 years ago. So what this method, how this method performs is basically what you do is you inject the DNA with interest into the lateral ventricle at a certain developmental age of the embryo. And then you use this type of electrode to deliver an electric pulse, which will pull the DNA of interest into the neuronal progenitor cells that's lining along the edge of the lateral ventricle. And then the neurons will be transfected or be transduced with the DNA of interest. And then you put the embryos back into the mom and wait for the pups to be born. So in our study, we are targeting layer 2-3 pyramidal neurons in the cerebral cortex. So we do electroporation roughly at E15. So a nice thing with this method is, depending on the time you do the electroporation, you can target different layers of the pyramidal cells. So give you some kind of flexibility in terms of studying which layer of neurons. So in our case, as I said, we are interested in the layer 2-3 population of neurons. We do in utero electroporation at E15. And then we wait for the pups to be born. When they reach young adulthood, we do craniotomy. Then we give the animal about 2 weeks to recover from surgery before we start 2-photon imaging. So another nice feature about this method is this is a construct-based method. So we can include multiple constructs at the same time. So in the following study I'm going to present today, we are transfecting sub-tag GLUA1, MIG-tag GLUA2, and a morphology marker DSRAD2. So the morphology marker DSRAD2 can not only help us to identify the neuronal processes, it can also help us to serve as a reference point when we do intensity analysis, which I'll briefly touch on later in the presentation. So first, when we use this method, first we want to see, okay, how efficient can we target those neurons, and how a good signal we can detect in terms of fluorine tagged AMPA receptor expression. So here what we're doing is, after in utero electroporation, we're doing an acute brain slice preparation at roughly P7, so with no immunostaining. After the acute brain slice, we just directly visualize under the microscope. As we can see, indeed, we can target a decent population of neurons with fluorine tagged AMPA receptors, and the expression of the AMPA receptor tag is pretty good. Again, next step is to do craniotomy. So for our case, we choose the open skull prep method. So you can either do open skull or thin skull. They have advantages and disadvantages of both. So the reason we choose open skull is we are expecting a relatively weaker expression of fluorine tagged AMPA receptors as compared to CERV-GFP, which is created by Ping some 20 years ago, have been widely used for two-photon imaging studies. Since the signal for the fluorine tagged AMPA receptor will be weaker, and then the open skull give you a much better chance in terms of detecting a weighted signal. So we choose open skull prep. So basically what you do is you cut off a piece of skull, replace it with a cover slip, and then seal with dental cement. So here I'm showing a blow-up picture. So as you can see, if you do a good job, you can see there's not much bleeding for the prep, and you can see very clear pattern of the vasculature. And also, this is how to say a relatively aggressive surgery. So after this surgery, we give the animal about 2 to 3 weeks to recover from the surgery before we start imaging under the microscope. So the first brain region we decide to start imaging AMPA receptor dynamics is the barrel cortex, or the somatosensory cortex. So the reason is there is previous study have been found that there is a very good one-on-one correlation between individual whisker and its corresponding barrel column inside the barrel cortex. So this one-on-one correlation adds some specificity to this system, made it a very attractive system to study synaptic plasticity. So on top of that, we can use intrinsic optical signaling to map out individual barrel columns. As I show you here, if you deflect one whisker, you'll detect a corresponding region in the brain. If you detect a different whisker, you'll detect a different region respond to the whisker deflection. So let me explain to you how what we are measuring for the intrinsic optical signaling. So here what we are measuring is the light deflection of the brain surface. What happens is if you deflect a specific whisker, the neurons in its corresponding brain region will be activated. then the local oxygen consumption will go up. So then the consequence of that is you'll have an increased concentration of deoxyhemoglobin. So then under wavelengths of 630 nanometers, the more deoxyhemoglobin you have, the less light will bounce off the brain surface. So in other words, if you deflect, the activated brain area will go stuck and then recover once the blood flow comes in and repopulate reoxygenate the deoxyhemoglobin. So using this method, we were able to map out the individual barrel columns. And then based on the position of the individual column and the pattern of the vasculature, we were able to zoom into a specific whisker and image the same neuronal processes again and again. So for the following study I'm going to present to you, the study was performed in anesthetized animals. And also we are deflecting one whisker at a time, and all the whiskers have been kept intact. So here I'm showing you a G-stack of the in vivo imaging of the SAP-GLUE1 we were able to acquire through our customized two-photon microscope through the cranial window. So here I'm showing you we are imaging from the top of the brain surface and going inside. And here basically give you an idea how deep we are in terms of relative to the brain surface. The green signal is from the SAP-GLUE1, and the magenta signal is from the DS-RED2. As you can see, as we go deeper, we can detect both decent signal from both channels, and as we go deeper, we can see all the neuronal processes converge to the cell body. And this is about 250 some microns deep into the brain surface. And here I'm showing you a 3D rendering of the previous Z-stack I just showed you. We can detect a cell body pretty well with a signal from both channels and sending beautiful neuronal processes to the top layer of the brain surface. And in the top layer, indeed, we can detect those green puncta-like structure, which are the SAP-tagged AMPA receptors. And another nice thing, as I just mentioned, those cell bodies about 250 to 300 microns deep into the brain surface, which are exactly layer 2-3 neurons should locate, which is our target population of neurons. So once we were able to detect a good signal in vivo for AMPA receptors, the next thing we want to see, okay, how variable is the AMPA receptor expression if you do no manipulation or whatsoever. So here I'm showing you, for this mice, we just go back to the same barrel column and image the same neuronal processes again and again for about a month. So with no sensory stimulation or any kind of manipulation. As we can see, indeed, over a course of 1 month, we were able to detect very stable expression in terms of the fluorine-tagged AMPA receptors and also for the DS-RED2. So another interesting phenomenon we notice from this experiment is that the relative synaptic strength judged by the intensity of the fluorine AMPA receptors is also kept relatively stable during this period of time. As you can see that for this spine, there's very strong AMPA receptor signal in the beginning. This one is always being the strongest in terms of AMPA receptor intensity, while for this one, there's relatively less AMPA receptors and has been kept stable during this 1 month period. So we think there is an intrinsic mechanism to keep the relative synaptic strength during this period of time, because the half-life of those AMPA receptors is about 40 hours. So those receptors or the intensity we see here at the end of the month is totally different from what we detect in the beginning. So there must be some kind of mechanism to cap or recognize the initial synaptic strength so that keep the relative synaptic strength stable during this long period of time. So once we acquire these nice images, the next big step is to do image analysis. Actually, I think for anybody who is interested or knows some basic knowledge about imaging is, one of the big step or one of the heavy step is to do image analysis. So luckily for us, another talented postdoc with David Linden, which is also at Hopkins, a floor down from us. So we were just having a casual talk during lunch. We're saying, okay, we are running into this nice problem. We have accumulated a big set of data. We are sitting there for us to analyze. So a thing with the software for the analysis, there are a couple of softwares out there, and it works okay for some immunostaining or dissociative culture. For in vivo imaging, actually, as of today, there's no automatic software or automatic program can do the job for you. So basically, he's also very interested in the problem. He's also doing some in vivo imaging. And then when we talk about it, we decide to do a custom design software. It's a semi-automatic custom. It's a semi-automatic software. The human beings still need to do a lot of the work. So I'll briefly walk you through how this software works. So basically, once you input those images into the software, what we do, the human beings need to do is to click the tip of the spine. Basically, tell the software, okay, here I recognize this is a spine. Then the software will automatically find the connection point between the spine and the dendritic shaft. Then once this connection is established, 3 regions of interest will be defined by the software. One region is the spine region of interest. This is about this square box. Another region is a background region of interest. Basically, it's in the neighborhood and to detect the background intensity. So another region is called the shaft region of interest. Basically, it's a connection point between the spine and the shaft, about 5 microns away from the connection point. So once this 3 region is defined, this 3 region will be automatically applied to both channels. So there are a couple of things we can analyze or we can look at for the images. First is a spine size. So the spine size is defined as such. So you calculate the intensity of the pixel in the spine region of interest, and then subtract the intensity of the pixel in the background region of interest, and then normalize against the intensity in the shaft region of interest. We define as spine structure intensity, which is a proximity or the estimate of the spine size. Of course, more interestingly, and the unique for our image study, is we are able to look at the intensity of the surface AMPA receptors. So there are 2 parameters we can look at. First is the spine surface AMPA receptors. Another one is the shaft surface AMPA receptors. I'll just walk you through the definition of the spine surface AMPA receptors. So it's the total intensity in the subgroup A1 channel in the spine region of interest, subtract the background region of interest, the total intensity, and then normalize against the dendritic shaft, which will be defined as the spine surface group A1 intensity. It's an indication of the intensity of AMPA receptors on the spine surface. So when we take a close look of the animals we electroporate, we were able to find that we were able to detect very beautiful signals at the tip of the spine. Another interesting phenomenon we find is that within a couple of microns from each other, we can detect dramatic differences in terms of their spine surface AMPA receptors. For this one, we get a lot of AMPA receptors in the spine surface, while for this one, we don't have much at all. So we speculate that for this type of spine, most likely represent the population of spines have active synapses, while for this population of spine, represents the spines with silent synapses. I have to admit that in our images, we were able to detect such phenomena, but especially in adult animals, those silent spines are very rare. So for the focusing study, for the following study, we are focusing on this type of spines, basically have some type of AMPA receptor to start with in our study, and then look at their dynamics after Whisker stimulation. So if we look at the whole spine population, we find that there is a wide distribution in terms of the spine surface AMPA receptors, as shown here. And also, interestingly, we find that under basal conditions, there is a very good correlation between the spine size, or the proxy of spine size, spine structure intensity, and the spine surface glutamate intensity. So indeed, previous studies have been using the size of the spine as an estimate of how much AMPA receptors is on the spine surface. Basically, our study kind of repeat their finding or are consistent with their assumption. While I'll tell you a little bit later, in terms of changes in those two, there are differences in our study. So as I briefly introduced to you that the first brain region we decided to study AMPA receptors is the barrel cortex. So what we are doing here, we want to see, okay, if we apply a rigorous, a very strong Whisker stimuli, what will happen to the spine surface AMPA receptors? So what we are doing is we are doing a 10 Hz 1 hour constant Whisker stimulation. What we do is we take images before we start the Whisker stimulation, then we start Whisker stimulation, then we take it immediately after the Whisker stimulation, and then 2 hours, 3 hours after the Whisker stimulation. So as shown here, if we look at this spine, for instance, we detect some type of AMPA receptors in the surface of the spine. After Whisker stimulation, we detect a dramatic increase in the amount, or based on the intensity of the surface spine AMPA receptors. This increase has been kept stable for at least 3 hours. While in the control group with no Whisker stimulation, as we can see for this spine instance, the amount of surface AMPA receptors don't change too much. So here is a quantification of all the spines we imaged. So overall, we imaged about 600 some spines in the stimulated group and 500 some spines in the control group. As shown here, if we look at the change of the spine size before and after Whisker stimulation, we don't detect much change. There is no difference between the stimulated group and the control group. However, if we look at the overall spine population, what we find is there is a dramatic increase in the spine surface AMPA receptors. As quantified here, they are about a 30% increase. And also, there is an increase in the shaft surface AMPA receptors. It's also about increase about 30%. As I said, so these three figures gives you an idea of when we look at the spine population as a whole, how does those spine surface AMPA receptor changes? But what does it mean in great detail exactly? So does it mean every spine change a little bit, or there's only a small population of spines change? And also, does it mean because in the neurons, every neuron has a couple of dendritic branches we can image, right? So does it mean different dendritic branches change the same after Whisker stimulation, or they're somewhat different, right? And also for all these three parameters, as I briefly introduced, in terms of the changes, are those correlated? Or what are the relationships between those changes? So those are the questions we dig a little bit deeper into the data, and I'll show you the result. So when we look at the spines individually, what we find is there's only about 30% of the spine show a change in the spine surface AMPA receptors after Whisker stimulation. The majority of spines don't show a change. This is also true when we look at the dendritic level. So for this figure, what we are doing here is, for one dendritic segment, we average all the spines in one particular dendritic segment and then assign a number. Then we compare that number before and after Whisker stimulation. So for the stimulated group, we image about 52 dendritic segments. While we find it's only about 11 of them, about 20% of the dendrite show an increase in their spine surface AMPA receptors after Whisker stimulation. So this tells us those changes in the spine surface AMPA receptors is not uniform across the barrel field. It's not uniform across even the dendritic segment. So there are only a subpopulation of spines and a subpopulation of dendritic shaft that respond to the Whisker stimulation. And also, as I said, what are the relationships in terms of looking at the changes of those three parameters? So here I'm showing you the correlation at 1 hour point after Whisker stimulation between the change of spine size and the change of surface AMPA receptors. Again, as compared to the baseline, there is also a decent correlation between the changes of those two. However, if you look carefully, we find that the average line is significantly above the y equal x line. What does it mean is, although there is a good correlation between the changes for these two parameters, but it seems like when we look at individual spine, the spine surface AMPA receptors always change more as compared to the change to spine size. Or in another word, after Whisker stimulation, we might detect a change in the density of the spine surface AMPA receptors. Here shows you that there is a very good correlation between the changes in the shaft surface AMPA receptors and the spine surface AMPA receptors. Those two parameters are very well correlated with each other. While on the other hand, there's no correlation detected between the change in the spine size and the change in the shaft surface AMPA receptors. So the take-home message from this is that the change in the spine surface AMPA receptors is very well correlated with the change of spine size and also with the change of shaft surface AMPA receptors. However, there are differences also between those three parameters. And for this one, I'll tell you a little bit later what are differences in terms of changes after Whisker stimulation. So the next thing we want to do is, what are the mechanisms, right? So we want to, here we want to briefly know a little bit more about the mechanism about this phenomenon. After Whisker stimulation, there is an increase in the spine surface AMPA receptors. What are the possible mechanisms? One of the usual suspect is AMD receptors, because AMD receptor-dependent LTP is the majority or the major type of LTP people have been studying. So we take an educated guess that let's first test whether this phenomenon is AMD receptor-dependent. So what we are doing here is we are applying a drug, a CPP, which is an AMD receptor antagonist. This drug can easily cross the blood-brain barrier. So what we are doing here, we are doing exactly the same procedure as the Whisker stimulation. The only difference is here we are applying CPP to those animals before we start Whisker stimulation. As indicated here in the representative images and also in the quantification at the bottom, the application of those CPP completely block the Whisker deflection-induced increase in terms of spine surface AMPA receptors. So if anything, there is a trend toward decrease, although statistically it's not significant. And also it blocks the changes caused by the Whisker stimulation for the shaft surface AMPA receptors. So basically what this figure tells you that indeed this process is an AMD receptor-dependent event. So as I have told you in the introduction, so another characteristics of LTP or LTD is its long-lasting, right? In the brain slice recording, it can last for about an hour or two, depending on how long you can hold the cell. So we took advantage of the imaging approach, and then we follow the AMPA receptors for even longer. Here I'm showing you for 48-hour time points. As shown here, again, there's no difference in terms of the spine size change before and after Whisker stimulation for short-term or long-term. For the change in the spine surface AMPA receptors, what we find is the increase of spine surface AMPA receptors after Whisker stimulation is relatively long-lasting. It can still have a dramatic difference at 48-hour time point. However, interestingly, the change in the shaft surface AMPA receptors comes back to about 6% at 6 hours, and then return to return to the level of the control at 48-hour time point. So that's what we are seeing here, because here we don't have the single molecule resolution yet. So we don't know exactly what's happening in terms of a single molecule. But what we speculate is that the insertion of AMPA receptors into the membrane surface and the incorporation of the AMPA receptors from the membrane surface into the spine are two related but also distinct processes and synaptic plasticity conditions. So here is the brief summary of what I've told you. First, we find that acute Whisker stimulation can lead to a rapid increase in the spine surface AMPA receptors, but only in a subpopulation of dendritic spines and a subpopulation of dendritic shaft. And also, I have shown you that this increase caused by Whisker stimulation is an AMPA receptor dependent process, and also is relatively long-lasting. So as I said, as compared to previous imaging studies, we add a new layer of plasticity to the study. So previously, people were just using the Cy1-GFP to image spine dynamics. And here, on top of spine dynamics, we can also measure the relative synaptic strength by monitoring the relative AMPA receptor intensity on the surface of the spine. So basically, our approach opens up a totally new way of imaging a synaptic plasticity. So next, I'll just briefly tell you a couple very interesting ongoing projects and what I'm going to do for my new lab. So as I said, the study I just presented to you were done in anesthetized animals, and we are using a artificial and a very strong Whisker stimulation paradigm. So okay, it's all nice, and we were able to observe those AMPA receptor dynamics in vivo for the first time. But really, what does it mean? We don't really know, because this is a very strong, a very vigorous Whisker stimulation the animal might never experience in their whole life. So the next, we want to understand a little bit deeper, or in another way, how does AMPA receptor behave under physiological conditions? Or how does AMPA receptor behave in awake behaving animals? Because when animal is under anesthesia, it's somewhat different in terms of their every parameter, right? So we observe this thing in the anesthetized animals. How does that will that transfer into awake behaving animals? We don't know yet. So that's why we want to further study the AMPA receptor dynamics during learning in awake behaving animals. So in collaboration with Assistant Professor Dan O'Connor at Hopkins, we developed this go-no-go task for the animal to learn or acquire, and then image while the animal is awake and performing the task. So what we do here is we restrict the water supply to the animal for about a week. So the animal is thirsty during this period of time, so it has motivation to search for water reward. Then after a week of water restriction, we put the animal under the microscope. So then we do a leaking period. What we do here is there is a metal pool within the reaching distance from the tongue of the mice. So if the animal leaks, then the metal pool will deliver a drop of water that the animal will get the water reward. So then we do 2 leaking sessions. Then what we do is we call it a discrimination. So then what the animal has to learn is once it's received a Whisker deflection signal, it's supposed to leak for water reward. If there's no Whisker deflection signal delivered, even if the animal leaks, there will be no water reward. So basically, the animal needs to acquire or get the sense that you have to receive the Whisker deflection signal before you leak or get the water reward. So on the right, I'm showing you a successful trial for the animal to get that water reward. As you can see, there is a glass pipette hanging in here, and a Whisker is poking into this glass pipette. What you'll see in the following video, this glass pipette will wiggle first. Then the animal get that signal, then it will leak for the water reward. So here comes the deflection, then the animal leak for the water reward. So if there's no deflection, even the animal leaks, there will be no water reward. So of course, in the beginning, the animal does not know what it's supposed to do. So it's basically guessing, or because it has the motivation to search for water reward, it's leaking most of the time. But half the time it's right, half the time it's wrong. So the performance is about 50%. But after 5 to 7 days of trial, you can see the performance of the animal in terms of the task is getting much better, jump to like 70%. And you can get this steady performance for a relatively long period of time. Interesting thing is, okay, once after about 2 weeks of training, we think, okay, this animal has successfully acquired this task. We just stop this training. What we do, we put the animal back into the home cage. We still restrict the water supply so that the animal is still thirsty. And then a week later, we put the animal back under the microscope. We find that the animal still remembers well about the task. Another week later, it still remembers. Another week later, it still remembers. So basically, what this assay shows you is that this is a very good paradigm, not only to assess how well the animal can acquire the task, it's also a very good paradigm to assess how well the animal can remember the task. So what we are doing now, we are imaging those animals in terms of, those are the electroporated animals with the fluorine-tagged AMPA receptors. So we are imaging AMPA receptors immediately after each and every training session. So we have acquired a rich set of data sitting there or in the process of analyzing. We want to see exactly in awake behaving animals under this learning paradigm, how does AMPA receptor traffic and behave during this learning and long-term memory task. So another direction I want to go is, as I said, we just briefly addressed what is a mechanism, right? You can say, okay, it's an easy guess. It's AMD receptor dependent. So what? What's a detailed mechanism? So in order to address that question, so a direction we want to go is it's a collaboration with Dr. Yong Zhang, who was previously at Hopkins, now recently moved to UCSD. She developed a series of kinase sensors that can monitor the kinase activity. So to this audience, I don't have to say too much. I just briefly introduce you. One type of sensors is FRET-based sensors. So if it's two fluorophores is far away, there's no FRET energy transfer. If after phosphorylation, under conformation change, those two fluorophores is close to each other, then there will be a FRET transfer. You shine light to one fluorophore, you'll detect the FRET signal. So here I'm showing you the experiment from the pKa kinase sensor. What we are doing here, this is the dissociative neuronal culture. This is the acute brain slice with a drug that can activate the pKa signal. We can see after treatment of the drug, there is a dramatic increase in terms of FRET signal. Basically, this shows you, indeed, this biosensor works very well in dissociative neuronal culture and acute brain slices. Do they work in vivo? So we also briefly test how well those sensors works in vivo. As shown here, those are the, again, transfected neurons. And here's the signal from the donor. Here's the signal from the acceptor. And we, using another paradigm, that epinephrine injection can also lead to an increase in the pKa kinase activity. There is an increase. Indeed, in terms of proof of principle, those FRET sensors can be used under in vivo conditions. And another thing we are in the process of doing that is also another type of biosensors. It's the single color biosensors, which can give us a capacity to monitor kinase activity or signaling pathway activity, multiple signaling pathway activity. As shown here, those single color biosensors also works well, as shown here. You see forscaling can lead to an increase for the pK sensor. BDNF can lead to an increase in the ERK sensor. And also, briefly in the dissociative neuronal culture, we're showing that forscaling is specific to activate pK, activate the pK kinase, and the DHPG can activate both. So, all right, here's a summary. In the future, we want to investigate AMPA receptor dynamics and the physiological manipulation conditions, and we're in the process of doing enriched environment and also Whisker trimming and such, and also to address the detailed molecular mechanism. We're going to use those FRET sensor or the single color biosensor in combination with the AMPA receptor imaging, trying to tease out the detailed molecular mechanism underlying synaptic plasticity. And also, we can use this method really to better understand our understanding of pathology or neurological disorders such as Alzheimer's disease. So first and foremost, I want to thank Rick Hugnier for his tremendous support, and thank all the members and the collaborators, and thank you for your attention. Thanks.